This second part of the screencast on organic molecules important to the human body covers lipids, or fats and oils. There are several reasons that lipids are necessary in the human diet. Our body uses them to build a very important component of cell membranes, phospholipids, and we will be talking about these phospholipids extensively as we move forward in the class. Additionally, fats can be used to supply energy for the body, as we discussed in the screencast on carbon. Because lipids are organic molecules, they contain carbon, hydrogen, and oxygen, just like the carbohydrates. However, they have a much smaller proportion of oxygen than do carbs. As an example of this, the most common type of lipid in the diet, a triglyceride, has a molecular formula of C55H98O6, which is a much lower proportion of oxygen than what is seen with the 2-to-1 ratio of hydrogen to oxygen in carbohydrates. There are several different types of lipids, but we will be looking at the structure of just three: triglycerides, phospholipids, and steroids. This first slide covers triglycerides, which are the most common type of fat in our diets. You'll notice in looking at the molecular structure of a triglyceride that it is a large and complicated molecule. They get their name from the fact that they have what's commonly referred to as a glycerol head portion, and attached to the glycerol head are three fatty acid tails or chains. So it's named a triglyceride because there are three tails attached to a head. For this reason, you will often see triglycerides represented as a little circle with three straight lines coming off of them, like you see here in the lower right corner of your screen. This is how I will represent triglycerides in notes and on exams and quizzes. As you look a little closer at the structure of the tails coming off of the glycerol head in the triglyceride pictured here, you'll notice that two of them are perfectly straight and the bottom one is bent. There is a reason for this, and it has to do with the carbon-carbon double bond in that third tail. Carbon is able to make 4 connections with other atoms all at the same time. You'll notice in the upper two tails that the carbon in the chain is bound to 4 other atoms in all cases. If carbon isn't bound to 4 other atoms, it will make a double bond to one of the atoms so that it still has 4 connections. In our bottom chain, the double bond is to an adjacent carbon, so it is a carbon-carbon double bond, and having this type of bond actually physically creates a kink in the chain. With some triglycerides, all of their fatty acid tails, or chains, have carbon that is bound to 4 other atoms, and in that case, their chains are completely straight. Triglycerides that have all completely straight chains come from saturated fats. In fact, saturated fats are referred to as saturated fats because of the fact that the carbon molecules in the chains are saturated, or making as many bonds to other molecules as they possibly can. Saturated fats tend to be solid at room temperature, think of lard or butter, and the reason they are solid at room temperature is that they can stick tightly together because they are completely straight molecules. For a visual of this, see the picture at the top right corner of the screen. Unsaturated fats are, as their name implies, fats that do not have all of their carbon atoms saturated or making bonds to 4 other atoms. Rather, unsaturated fats have 1 or more carbon-carbon double bonds in their tails. Because these bonds cause kinks, unsaturated fats tend to be liquid at room temperature, as their molecules physically cannot stack together as tightly or densely. Again, if you need a visual of this, see the picture in the top right corner of your screen. Olive oil and fish oil are good examples of unsaturated fats that are liquid at room temperature because their molecules cannot stack tightly together. You've probably heard of monounsaturated and polyunsaturated fats, and that just has to do with whether there is only a single carbon-carbon double bond or more than one. Monounsaturated fats, such as olive oil, have just a single double bond, while the polyunsaturated fats, such as fish oil, have more than one. You've probably also heard of omega-3 and omega-6 unsaturated fats, and these names are just describing where along the fatty acid tail the double bond occurs. With an omega-3, it is in the 3rd bond, and with an omega-6 in the 6th bond between carbons. You've probably also heard that unsaturated fats are considered to be the more healthy type of fat. The reason for this also goes back to that carbon-carbon double bond. Believe it or not, a double bond between carbon atoms is actually less stable and more easy to break than a single bond between carbon and hydrogen. The reasons for this are due to organic chemistry that is beyond the scope of this class. However, because of this, unsaturated fats are broken down and processed by the body quicker and easier than saturated fats. Saturated fats tend to stick around longer in the blood, increasing the risk of cardiovascular disease. And that brings us to the worst fat of all, trans fats. Trans fats start out as unsaturated fats. What happens is we take an unsaturated fat, in this case vegetable oil, and we try to saturate the carbon with hydrogen atoms. Basically, they try to break the carbon-carbon double bonds and replace those connections with connections to hydrogen atoms. This process, where we basically add hydrogen to an unsaturated fat, is known as hydrogenation. So if you ever notice the terms hydrogenated or partially hydrogenated on the list of ingredients in a food you are eating, that food has trans fats in it, whether it is listing them on the nutrition label or not. So why would you want to break double bonds and replace them with single bonds to hydrogen? Here's why. Remember, those carbon-carbon double bonds are not very stable. They break down easier and more quickly, and so products that contain unsaturated fats are not as shelf stable as products that contain trans fats. If we can convert an unsaturated fat into a trans fat, then put the trans fat into food, the food will have a longer shelf life, and that is why trans fats are so common in processed foods like crackers that may sit on a shelf at a store for a long period of time before being purchased and consumed. The problem with trans fats is that the hydrogenation process is never completed, so you end up with a fat that is no longer an unsaturated fat, but it is also not a saturated fat. It is somewhere in between the two, an unnatural molecule that our bodies don't recognize and don't know what to do with, and that really causes problems for us, and especially for the cardiovascular system. I mentioned phospholipids at the beginning of this screencast, and that they are important parts of the cell membrane. Phospholipids are similar in structure to triglycerides. If you look at the first picture here at the bottom of the screen, we've got a triglyceride with a glycerol head and 3 fatty acid tails attached to it. A phospholipid has the exact same structure, except for that one of the long fatty acid tails is replaced with a short phosphate group, or a molecule that contains phosphorus. You can see the molecular structure of a phospholipid here in the center of the picture. I want you to focus for a second on the fact that the phosphate group is charged. There is a little oxygen atom with a negative charge on it, and that makes the head of the phospholipid molecule charged. You may remember that water is a molecule that is also charged for reasons that we've talked about previously. Because the head part of the phospholipid is charged, it is attracted to other charged molecules such as water. This makes the phospholipid head hydrophilic or water-loving. The heads of these phospholipids like to be in a watery environment. The tails are not charged, so they do not like to be in a watery environment. In fact, they hate being in a watery environment and will avoid it at all costs. For that reason, the tails are referred to as hydrophobic or water-hating. The fact that phospholipids are hydrophilic on one end due to the charged oxygen and hydrophobic on the other gives them some pretty amazing abilities that we will get into more when we talk about cells and the cell membrane in particular. So for now, just kind of burn that idea into your head because it becomes very, very important for understanding cells later on. The last part of the picture on the bottom right of your screen shows a representation of a phospholipid. Phospholipids are often drawn similar to the way that triglycerides are drawn, but because they only have 2 fatty acid tails instead of 3, they look a little different. Keep this image in your mind because this is how I will draw phospholipids on exams and quizzes and also in my notes. The last type of fat I want to mention just briefly. Steroids are lipids with a complex interconnected ring structure. Due to their complicated structure, they tend to be rather rigid molecules. We find steroid-based molecules, or molecules that have structures based on or similar to steroids, throughout the body. Cholesterol is a steroid-based molecule which is pictured here. Several hormones are also steroid-based, including the sex hormones, which are estrogen and progesterone in females and testosterone in males, and some of the hormones that are produced by the adrenal glands, which we'll talk about towards the end of the course when we get to the endocrine system. This completes the screencast on lipids.